Good afternoon, good evening, almost everyone. Welcome to this month's Thursday Botsok Conservation webinar. My name is Martina Trienicht. I am Botsok's Conservation Project Manager, and it's my pleasure to welcome you all here today, members, volunteers, and anyone from the public. I'd like to ask our three guest speakers to switch on their cameras. Before I introduce them, I'll introduce them to everyone shortly. But just a quick announcement for making the webinar run smoothly, and just a few reminders also. You are welcome to post your location in the chat. It's always nice to see where people are dialing in from. And we'll have three presentations. First, we're going to have Ian Patterson, and then Kim Weaver and Nompumelelo Bazo. I'll introduce their titles also to you shortly. They will give their presentations, and you are welcome to ask them questions in the chat in between the presentations or during the presentations, and we'll try and see how many of your questions we can answer also directly. Please note that the webinar is always available for review on the Botsok YouTube channel. So anyone that isn't able to join us live can recap it there, or just in general for afterwards. If there was something interesting, you can of course refer to that as a resource. All right. So our first speaker that I would like to introduce to you is Professor Ian Patterson. He is the Deputy Director of the Center for Biological Control at Rhodes University. Ian has done lots of research on aquatic or biological control of weeds. And I believe Ian has many students that is doing research for him or on his behalf and with him in the field. Then our second speaker is Kim Weaver. Kim Weaver is more involved in the outreach or community aspect of biocontrol, and she is specifically a community engagement officer, also based at Rhodes University. Kim's talk will revolve or is titled Promoting Biological Control, which leads to improved implementation and long-term sustainability. So Kim, we look forward to your talk after Ian. And then our third speaker is Nompumelelo Bazo. Nompumelelo is a postdoc from the South African Institute of Aquatic Biodiversity, and Nompumelelo will be sharing her journey as a scientist in the biocontrol space with us as the third speaker. So I think from my side, that's a general introduction and just setting the scene for the day. I thought because the topic of biocontrol is perhaps not so familiar to us, and I should mention the title of the webinar also, which I haven't. So we are all talking about balance today, balance restored, biocontrols for biodiversity as a tool to safeguard or to promote biodiversity in South Africa specifically, but also in different ecosystems. And I thought to start just by setting the scene, asking firstly, Nompumelelo, for you as a relatively early career researcher, where did you become interested in biocontrols, and what was the sort of moment for you where you realized this is the field that I would like to specialize in?
Hi, Martina. Thank you for the introduction. I think the biocontrol bug bit me in third year, to be specific. We had a lecturer by Professor Julie Kotsia, who's been my academic mother since then. So she was talking about invasive species and the different types of ways in which they are controlled, especially in freshwater ecosystems. And there was three options that she introduced. It was mechanical control, manual removal, and biological control. And so during the time of her lecturing, I saw I saw that biocontrol seems to be the better option because you know how chemicals we don't want them in our waters, and manual removal seem to need more like resources. So that's where the bug bit me, and then I've been in it ever since.
So the bug did bite you.
Yes,
Thank you,
yes, it did.
Nompumelelo, for sharing that. Okay, then for Ian, maybe, as a specialist in this field, also from a research perspective, not also, but what in your view would you say makes biocontrol so different from, for instance, spraying or chemical methods of alien control, which is obviously a big problem across the world, but especially also in South Africa?
Good evening, everyone. Thanks, Martina. I think the most important difference with biological control is that it is safe and sustainable. So it's safe in terms of we're not having to spray harmful chemicals into our ecosystems, which are harmful to both humans and to other organisms. And then the other thing is the sustainability. It's really the only way that we can permanently reduce the negative impacts of any invasive alien plants. And I'll be talking a bit about that in my talk that's coming up.
Mm, Thank you, Ian. I'm sure you will. Kim, and then for you, more from the community perspective, sometimes it might not be easy to explain to communities or to people how to fight or how to use an insect, to fight a weed or a plant, and whether that's actually a good thing. So what would you say, how do you find people generally towards this topic? And yeah, are they relatively open or a bit more skeptical?
Yeah, I think there's a bit of both. But overall, I think if we explain it properly and if people understand the science behind biological control, then there's huge support for it when people can see it working and when they understand how it works. So that's really where all of us work in this space. We need to ensure that people understand it so that they can support it and accept it as a control option. Yeah.
Okay, I look forward to, I think we'll unpack more of these concepts, obviously, as we go through the presentations and your talks. So Ian, I think on that note, I'm going to ask Nompumelelo and Kim to switch off their videos, and we'll see you shortly. And give Ian a minute or a moment just to share his slides. And yeah, looking forward to your talk then, Ian. Thank you.
Thanks so much, Martina. Okay.
Just go full display, Ian. Sorry about that. There you are. Thank you.
Cool. Yeah, good evening again, everyone. So my talk is entitled Biocontrol for Nature. And I really want to talk about what biological control can do to control the huge problem that we have with alien invasive plants in South Africa. And this first slide has a photograph with two hippos. These hippos are not happy hippos, unfortunately, because their ecosystem has been clogged up with an invasive alien plant that comes from South America. This is water hyacinth. And there are very few floating aquatic plants that are similar to water hyacinth that are found in the continent of Africa, and they never get nearly as abundant as you can see in this photograph. So under normal circumstances, this would be an open body of water, there would be light penetrating the water, and the ecosystem would be a lot healthier and better for the hippos to live in. This is a photograph taken in the fynbos biome, somewhere near Cape Town, and just about everything that you can see in this photograph is actually a plant from somewhere else in the world. So what we should be seeing here is a really diverse amount of fynbos vegetation. We would see very few large trees in natural fynbos. But what we do see is just a small number of very, very abundant plants from elsewhere in the world. In the actual river, in this photograph, it is all clogged up with a plant from Australia. It's one of the Australian wattles. On the bank from where the photograph was taken, that really bright green stuff that's starting to come up, that's new growth of Sesbania perniciea, which is a plant from South America. The large grey trees are eucalypts that are also from Australia. And then if you get into the valleys leading up into the mountains, you'll find that they're clogged up with pines that come from Europe. So instead of finding lots of a high diversity, lots of different species of indigenous fynbos plants, we see that most of this photograph is taken up by just a handful of plants from elsewhere in the world. And these plants have changed the functional composition of this ecosystem. As I said, we normally wouldn't have big trees like this, that as abundant as this in the fynbos biome. We also wouldn't have the whole river clogged up with large shrubs. So this changes the way that the ecosystem functions, and it changes the services that the ecosystem provides. So we know that alien invasive plants have negative impacts to ecosystem services, and we know that this actually costs South Africa a lot of money. One person who's done quite a lot of work on this topic is Brian van Wilgen, and I've taken these graphics from one of his papers. This paper focused on just the grassland biome of South Africa, and they looked at the amount of water that's produced basically by that biome and the amount of heads of large livestock that can be kept in the biome. And they worked out how much water and livestock would be able to be kept there without invasive alien plants. Then they looked at what can actually be kept there at the current situation with the level of infestation of invasive alien plants in that biome that we have today. And that's shown in the gray bar. The white bars are if there were no invasive alien plants. And then the really scary one are the darkest bars, which are what would happen in 50 years' time if we don't do anything about the invasive alien plants and let them increase at the current rate that they are increasing. So you'll immediately notice it looks like there's not much of a difference between when there are no alien invasive plants and our current situation. But we're dealing with very big numbers here. So for water, that's 1 billion meters cubed of water less that is produced by the grassland biome because of invasive alien plants at the current level of the infestation. So that is a very large amount of water. It's about 190,000 heads of cattle less that we can keep in this biome because of the impacts of invasive alien plants. But much more worryingly is what will happen in the future. We'd have a collapse to about half as much water as we get today from the grassland biome, and only about 20% of the number of cattle would be able to be kept in that biome. This would obviously have huge social implications and financial implications in the country. Overall, the estimates of the costs of invasive alien plants to South Africa in terms of the reduction of ecosystem services that they provide is about 6.5 billion rands per year. So this is a really huge problem, and we need to do something about it. So one of the solutions to the invasive alien plant problem is to use biological control. And just to explain what biological control is, I've got this little schematic diagram here. You can see that there's a plant in the indigenous distribution. And so for a pine, that would be from Europe. For an Australian acacia, it would be back in Australia. It's where the plant evolved and where it is indigenous and belongs. There are always natural enemies that are feeding on these plants in the indigenous distribution. By far the most important natural enemies of plants in nearly every case are herbivorous insects and fungal pathogens. And of those, the most important natural enemies are the specialist natural enemies. Only about 30% of the natural enemies of a plant will be generalists. By generalists, I mean that they can feed on that plant and a few other plant species, or quite a lot of other plant species. A specialist is one that will only feed on that plant species or a few close relatives of that plant. And the vast majority of herbivorous insects and fungal pathogens are in fact specialists. About 70% of all of them are specialists. So those specialists are really what regulates plant populations in the indigenous distribution. When we bring these pines over from Europe and the acacias over from Australia, we leave those specialist natural enemies behind. And that means that the plants here in South Africa have a competitive advantage over all of the indigenous plants, which have their own specialist natural enemies feeding on them. And that's really one of the main reasons why alien plants become invasive and overabundant and problematic. So what biological control does is we get one of those specialist natural enemies, or maybe more than one, and we introduce it where the plant is alien, and it will reduce the negative impacts associated with that invasive alien plant. Now, the trick here is obviously that biological control agent, that natural enemy, needs to be host specific. It needs to only feed on this particular plant and not feed on any of the indigenous plants in the country or any of the crops or other valued plants that we don't want damaged in the country. And most of a biological control scientist's time at work is actually spent answering this question and finding out whether natural enemies can be used safely as biological control agents. It takes somewhere between 5 and 7 years of a scientist's career to determine whether a natural enemy is safe for release. And we know that once we've done this, it's a very standardized protocol that's been very well studied. And we know that once we use this protocol, we can accurately predict what will happen after the biocontrol agent has been released. And we have some evidence for this. So biocontrol is nothing new in South Africa. The first biocontrol agent against an invasive alien plant was released way back in 1913. So we have 120 something years to look back on. And in that time, we've released over 130 biocontrol agents for over 70 weeds in South Africa. And of those, absolutely none of them have gone on to do anything that was not expected during the host specificity testing, during our pre-release testing. So none of them have gone on to feed on indigenous plants, damage indigenous plants, or damage valued crops in the country. So it is a safe practice that is not harmful to our ecosystems. This is just a photograph that illustrates what host specificity can look like in the field. This is a population of jointed cactus, a terrible invasive plant from South America that had a big insurgency in the northern parts of the Kruger National Park. And we took the biocontrol agent there. It's a species of cochineal insect. It's that fluffy little ball that you can see in the inset photograph. It feeds on the cactus and injects a poisonous saliva into the cactus, and it produces that fluff to protect itself from predators. All of the stuff lying down on the floor in the middle of that photo is dead jointed cactus that has been killed by this biocontrol agent. But when the cactus dies, the agent will die because it can only feed on that cactus. It can't feed on any other plants that are present in the Kruger Park, or in fact anywhere else in South Africa or on the African continent. So those very similar looking euphorbias, which are often mistaken for cacti, cannot be fed on by this insect at all, and they are perfectly safe. So I'd just like to run through one or two biocontrol programs that have been successful in South Africa so that you can get an idea of what the benefits of biocontrol can be. So the Kruger National Park was once infested with this plant called the Puntia stricta. It's a cactus species again, and it is from Texas in the USA. And at one stage, 30,000 hectares of the Kruger National Park were covered with dense infestations of this cactus species. And you can see that that cactus is covered with fruits. Every one of those fruits is packed full with seeds. Just to clarify, that 30,000 hectares is of the dense infestations. The area that was infested by this plant actually was about 66,000 hectares within just the Kruger National Park. So this was an ecological disaster. The baboons and the elephants really like eating the fruit, and that makes the problem much worse because you can see in the droppings there that it's packed full with seeds, and that spread the plant all over the Kruger Park, and it became really, really aggressive and abundant. Thankfully, we have a very effective biological control agent for this plant. It is another species of cochineal insect, and this species can only live on this species of cactus. And you can see the kind of damage that it does. So in the photograph of the insect there, you can see the fluff that the insect exudes. And then that little insect where you can see its legs in the front of the photograph is a crawler. That's the stage of the insect's life that it disperses with. And then it becomes sedentary and sucks out the juices of the plant and injects that poisonous saliva. And you can see the before and after photos taken in the Kruger Park of what it looks like when the plants get damaged by this cochineal insect. So some monitoring was done in the Kruger National Park, and this is just the data from 1992 till 2014. The dark line is the abundance of the cactus, and the dotted line is the abundance of the biocontrol agent. And you can see that before about 2001, there were quite high densities of the biocontrol agent. So it says cladodes per meter squared. A cladode is simply a pad of the cactus. So there were somewhere between 20 and 35 pads of cactus per meter squared in the Kruger National Park, in the areas where the study was done, before the biological control agents. That's really a lot of cactus. You can see there's a bit of a fluctuation from 1992 to 96. It's lower than from 96 till 2001. That's probably because it was wetter from 96 till 2001, so the cactus did a bit better in the ecosystem. The cochineal insect was released in 1997. At first, the scientists who released it were very worried that it wasn't exploding, but cochineal doesn't really like wet weather. And when it started to get dry, the cochineal exploded. So you can see in 2001, there's a massive increase in the abundance of the cochineal insect and an associated decline in the abundance of the cactus. The important thing here is that after 2002, it had been reduced to about 3 or 4 cladodes per meter squared, which is much lower and much less harmful to the ecosystem. The really important thing is that it's been kept at that low level permanently. So that's the sustainability of biological control. Once we get control, it is permanent. So this has been hugely beneficial to the Kruger National Park and to the indigenous biodiversity of South Africa. So just to show you what this looks like, that's a photo taken before biological control. You can see those 2 little lines on the tree in the background. This photo was taken years later in exactly the same place. The tree's grown a little bit, but you can still see the lines on it faintly. And there's hardly any cactus there, but there is a little bit of cactus. There will always be some of the weed left when we use biological control. But every time the cactus population increases, the Asian population will increase following it, and it'll be kept at this very low level where it is no longer a problematic weed. Biological control has also been very effective on water weeds. This is Azolla folliculoides, which is a floating fern that comes from South America. And this is a water body near my hometown in Makanda. And it covered this water body and many other water bodies around the country. The mat of the fern is about 1/2 meter thick in this case. All the water underneath it goes really smelly and low in oxygen. Things really struggle to survive underneath it. This site in particular, where this photograph is taken, is a very important site because it's a culturally significant site where there were ceremonies and spiritually important ceremonies. It's a place where local people connect with nature. And when the water body was covered, it was spiritually closed for business. It's also the only known site in the whole world where a fish called Sandelia bainzii, which is a small species of minnow, is known to reproduce. So it's the only known breeding site of a critically endangered fish species. A little weevil called Stenopalmus was released as a biocontrol agent. That weevil obviously came from the indigenous distribution of Azolla fern. It can only feed on Azolla. And after it was released, within 6 months to a year, every site started to look like this. So there's still a tiny bit of Azolla left around the edge of the dam, but it's permanently controlled it across the whole country. And you will hardly ever get sites where Azolla will even cover the half of the impoundment. And the benefits to South Africa from this have been absolutely huge. It's opened a culturally important site again so that people can now utilize this area and connect with nature. It almost certainly saved a critically endangered species of fish from extinction. And when a cost-benefit analysis was done from farmers all over the country, they found that just a few years after the agent was released, in the year 2000, they found that for every 1 rand that was put in the project, farmers were already getting 2.5 rands back. 10 years later, that was 15 rand for every 1 rand. For now, it must be 50 or 60 rand for every 1 rand that was put in. And that's because it is a sustainable method where the control is permanent. So just to come to a conclusion here, the main messages from my talk are that biological control is an environmentally safe practice, and it's founded on good, solid science. It's a long-term solution. It can take quite a while before it is implemented, but the benefits are permanent, and the benefits to biodiversity are permanent. In South Africa, we have 87 biocontrol agents that have been established on 66 target weeds. So some of those biocontrol agents that I talked about in the beginning of the talk haven't established, or we don't know that they're established yet. We can only do post-release evaluations about 10 years after the release of most biocontrol agents because it's a long-term intervention. And our assessments show that about 30% of our biocontrol agents have had what we consider a minimal impact. So they haven't controlled the weed completely, but they've done some good. About 42% of them have had a substantial impact. That means that the ecosystem services that were being stopped by the biocontrol agent have to some extent returned, And also, we are better able to control those plants using other methods like mechanical and chemical control. And in 28% of those cases, there's been complete and permanent control. And the benefits from when we get that are just so huge that it's really, really worth striving for. And those are situations like the two examples that I've given you in this talk, of which there are many others. So the Centre for Biological Control, where I work, is always working on developing new biocontrol agents, because we need a new biocontrol agent for each new weed. Because they are host specific, you have to have a tailor-made agent that is a solution to each different weed problem. And that's what the Centre for Biocontrol spends a lot of its time doing, is developing new biocontrol agents for new problems to protect this wonderful biodiversity of South Africa, like what is shown in the photo here. With that, I'll end my talk. Thank you so much for your time. It's wonderful to be able to talk to so many people who love the indigenous biodiversity of South Africa. Thank you.
Thank you, Ian. Thank you for kicking us off with such a nice talk, and for, setting the scene and also highlighting some of the successes that have come from biocontrol. Ian, I don't see any immediate questions in the chat for you, but I'm curious, and only because just from a curiosity perspective, you mentioned that the development of an agent for biocontrol takes about 5 to 7 years. Now that is a reasonably or relatively long time in measured in human years at least. But can you explain a little bit more to us just about what that process looks like? So, which components would come in? I'm assuming maybe lab components, obviously technical side, and then also field trials to actually get to the point of where you say this is potentially something that we can use in the field.
Yes, sure. So we have to, first of all, get out to the indigenous distribution and find what's available. In many cases, nobody knows what species of natural enemies are regulating these weeds in the indigenous distribution. And ironically, sometimes these plants are very difficult to find. They're sometimes very rare. And in some cases, they're even endangered in the indigenous distribution. Then we need to find those insects. We need to bring them back to South Africa, where we keep them under quarantine conditions, which are very strict conditions where the agent, the insects or fungal pathogens can't get out before we study them and know that they're safe. We then test them on a whole lot of species that are closely related to the target weed, because a natural enemy is, if it can feed on something else, it is likely to feed on the close relatives of the plant that it's found feeding on, rather than more distantly related plant species, because all the chemical cues and the nutritional value and the actual structures of the plants are going to be more similar in close relatives. So we test all the close relatives, and then we test less and less representatives of more distantly related taxons that are important, until we've described the whole host range of that potential biocontrol agent. Then we have to write up applications that go to the government, to various different government agencies who follow our recommendations and make the decision as to whether this is something that the country wants as a whole.
Thank you. Yeah, quite an intensive and a rigorous process, and lots of hoops to jump through, I'd imagine.
Yes, we have to be very, very careful in doing this, because the unintended consequences, if we get it wrong, are permanent, just like the benefits are permanent. So we do it very carefully, but our track record shows that we have been doing it carefully and that we do know how to do it properly. There was a question about lantana. Yes, there are biological control agents for lantana. They probably fit into the category of not fully controlling the weed, but they do quite a lot of good on a landscape level. And there's several of them.
Thank you. So I think if you would like to, Ian, then you can outline some of them in the chat in response to that question about lantana. And then, yeah, we're going to thank Ian and invite Kim to switch on her camera as our next speaker. Kim, thank you, Ian.
Well, can I go ahead and share?
You can go ahead and share, Kim, and we look forward to your talk a bit more from the people's perspective on this topic. Thank you.
Great. Thanks so much for having me here this evening or this afternoon. Yeah, so as Ian has introduced biological control as a concept very nicely and introduced our science. And now we will talk about how we promote biological control so that we can get the public support in it, and which will improve our implementation and our long-term sustainability. Yeah, so the responsibility of promoting awareness of biological control has primarily fallen on the researchers and the students associated with each program. Implementing biocontrol has slowly shifted to the researchers as competence and capacity has dropped within the government structures. This is done through our researchers participating in community engagement activities. And as a research center based in a university, our core focus is research, although we do quite a lot of implementation too, which I'll get to in a bit. But how do we get our research to reach the public? And this is through engaging with our communities. So our community engagement activities at the Center for Biological Control are guided by creating general awareness of invasive species and biological control, and bringing people's attention to the center as well as the university. Part of our research is researching the impact of our biocontrol programs, and for this we need sites that landowners and land managers can assure us that no herbicide use and no manual removal occurs. This helps us evaluate the impact of our biocontrol agents over time. And we really do need the support of communities and governments so that they can assist in implementing biocontrol. And I'll get to this later in how we have worked with community partners in the actual implementation of biocontrol. And obviously, feedback is essential, and especially to the people that fund our work. Over the years, it's moved more from government funding to other sectors funding the work. And yeah, and we do this through engaging with different stakeholders through the different platforms that we use. And ultimately, we really want biocontrol to be implemented wherever possible. So we really need more people to push this agenda with us. Yeah, so how do we raise awareness in the hope to foster a culture of environmental stewardship? So we do this through various social media platforms and sharing our news. We create posters, popular articles of our research, and especially our wins, which Ian has shared some of them, and our partnerships that we foster through the work that we do assist us in getting information out on their various platforms too. And our researchers use a lot of iNaturalist. In the last couple of years, we use this a lot to record and share locations of invasive species. iNaturalist has allowed us to find new sites where we didn't know the invasive species was located yet. Yeah, and here are some examples of some of our popular articles. Obviously, you all know about Hartbeespoort Dam and then the Vaal River recently. Yeah, so we try and get as much out there. We've created some awesome cartoons where we can explain the science in a very accessible way for people to understand. So I'm going to speak mostly about our mass rearing for and with the community engagement program. But yeah, I'd also like to share some of the other programs that we run. Yeah, we really enjoy sharing our science with schools. We find young minds are often more accepting of the science and understanding, and we hope that this goes home with the learners too. And yeah, we always try and do outreach with young people when it is possible. We run an annual accredited short course through Rhodes University and have done so since 2005. The course has had over 150 delegates from over 17 countries. Our next accredited short course is in November, this year. Starts on the 17th till the 20th, where we are going to welcome 26 participants from and 3 different countries represented amongst those participants. There's a huge demand for this sort of training, and we are hoping to possibly even do 2 courses next year. And then training, informal training. We do a lot of informal training with our partners, our community partners, and this is just responding to needs and responding to things that they want to know. And then our Sesonke program, this is where we upskill and employ people living with disabilities to rear biocontrol agents. We have 15 people currently employed that are living with disabilities in our rearing programs in the country. And yeah, we really, because our history with our funding is through the Working for Water program, obviously this was a big call to employ people living with disabilities, and we really do see the value and enjoy working with some of the underprivileged people in our country to make a difference in rearing and implementing biocontrol. So mass rearing is essentially the farming of insects to use as host-specific pests on selected invasive species. And we ensure that the releases are done with the correct insect on the correct problem plant. As Ian shared with us earlier, each insect is host specific, so can only work on the intended weed that it's meant to work on. Through our teams working hard at rearing insects, we can maximize numbers of insects going out to sites that need them. And this is not only in South Africa. Our insects are well traveled and have traveled throughout the globe. The CBC also collect and redistribute biocontrol agents that cannot be mass reared. These are mostly on woody weeds, so Sesbania, Australian acacia. And we have a new program on this that we are trying to do more collection and redistribution of biocontrol agents. Yeah, and our rearing efforts have made a considerable difference to these biocontrol programs. Water weeds throughout South Africa, water weeds plague our water systems. And obviously the real problem is the eutrophication of the water, but we are fighting the weeds, so that is what our job is here, and I'll share more about this later. And cactus species, of which Ian shared a few examples with earlier, and Naltuma or Prosopis or mesquite, commonly known as, which plagues the Northern Cape and our drier regions in South Africa. And yeah, the aim of each biocontrol program is for biocontrol agents to establish by releasing enough agents. So yeah, you can't just release 2 insects and hope that the population will establish and make a difference. So each insect has a different number that we like to release, so that we can ensure or hope that the insect establishes. And we do post-release evaluation to ensure that they do establish. Awareness to be raised about the plant and associated insects. So some plants have more than one insect that control it. Community partnerships to form to increase the public participation. So here's a great example of public participation with the waterweed mass rearing. We have a facility here in Makanda, in my hometown. And we have 20 community-run facilities that we support and assist where possible. But a lot of the resources are put up by the community partners. And yeah, this is really, we have a postdoc fellow who sees to this program in the Highveld, and she goes and visits these stations almost monthly or sometimes even twice a month. And these community partners are really making a difference in releasing agents on their systems that they are affected by. And cactus mass rearing, we, as the center, run a facility in Karicha, or Uitenhage, as it was previously known as. And we have 2 community-run facilities that are rearing cactus biocontrol agents too. And a newer program that has been established over the last 2 years, which is completely run by a community partner, which is the Association of and for Persons with Disabilities in Uppington. And they are rearing cactus biocontrol agents as well as insects for mesquite, and releasing them in the field. Yeah, and the characteristics contributing to the success of these biocontrol programs are over 2 decades of sufficient funding for implementation of biocontrol. And we still have sufficient funding for the implementation of a lot of our programs. Competent capacity in our research and implementation teams, community partners who share our goal and implement with us, weeds which people want to get rid of and that have good working biocontrol agents, sites that are iconic and economically important. So these invasions put us on the map, such as Hartebeespoort Dam and the Vol River. Yeah, we raising awareness through public talks, popular articles, and social media presence. This all helps the success of our biocontrol programs. So even us talking here today. Yeah, and through our activities with our community members, we have enabled participation in social learning spaces. And yeah, we've looked at this learning in these spaces and how so much value is created for the participants that are involved in them, as we all strive to make a difference with the shared objective of fighting invasive species in South Africa. And through involving the community members and forming these partnerships, it has resulted in more biocontrol agents being reared and released in South Africa, new invasion sites being reported, more post-release evaluations conducted, which is really important so that we can see the impact of our biocontrol agents, and for more funding received, which has been a huge help since our government funding has not been as forthcoming. And yeah, community members working together towards the shared goal. So in conclusion, community engagement efforts raise awareness with the general public as well as decision makers, so it helps put our research on their radar. Participants find value in making a difference and working towards the shared goal. And yeah, maintaining capacity in biocontrol has been very important. And yeah, research institutes and community partnerships are important to sustain this capacity as well as sustain the biocontrol programs. So the way forward for the center is receiving ongoing support to community partners. So we giving ongoing support to our community partners through regular visits and responding to training needs. And we really encourage and support the release of biocontrol agents where needed. And we are starting to work with government departments. Well, we always have, but we reformalizing these, working with government departments to use and implement biocontrol. And yeah, we're having more strategic and planned awareness campaigns so that we can get our research out there and hopefully have a higher uptake. Cool. And with that, I'd like to just say thank you for listening to me. Yeah, and if there's any questions.
Thank you, Kim, for that excellent work that the center is doing with communities and all the different groups, really making a difference out there on multiple frontiers. So a very nice overview. I see we've got a couple of comments just from Tracy, who's saying she's very grateful for a starter kit that she got for a wetland forest ecosystem. So just to reiterate, Tracy, we see your comment and glad that you've received that. There's another comment or a question rather. So would it be possible to distribute the presentations to the participants? So as BOTSOC, we don't distribute the presentations, but you're welcome to contact the speakers, and we will leave it on their side to decide. But also just to say that the webinar is available afterwards on the YouTube channel for anyone that would like to review some of the talks or share it with other people. Then you can share the webinar through the YouTube link. And that should be available in the next 2 to 3 days. And just a kind request, I see there's a couple of AI note takers also in our meeting, and I would kindly just like people to request to remove their note takers, and that we be respectful of our speakers and presenters today. Thank you so much. Kim, so yeah, not a specific question, but maybe just highlighting again the main age group. You mentioned that you have 15 people employed in your programs. And is your age group that you're focusing on, is it mostly 18 to 35? So in the standard youth definition, or do you also work with school groups?
In terms of our employment, so we employ more than 15 people. It's just 15 people living with disabilities in the Sisonke program. But no, we our range, we have employees of all ages in our research group and our implementation group. And the school work that we do is just more the awareness and trying to share our science. Yeah.
Okay, thanks, Kim. Then I'm just going to flag 1 or 2 more of the questions so that we stick to time. There's comments. Thank you for the
I can
great
respond
work.
in the comments if you want.
You can respond in the comments, and if you could just put for David the name of the postdoc, perhaps that he can contact the postdoc that you mentioned in the Highveld.
Perfect.
Thanks. Thank you very much. Nompumelelo, so at this point, you're welcome to switch on your video, and we look forward to your talk. Thank you very much, Kim. Appreciate that.
Is that coming in fine?
That is, thank you.
Thank you. Hi, everyone, and thank you for joining us. So I feel Ian and Kim have both set a good background for me, so I won't spend as much time on the technical side. So I'm just going to tell my personal story and personal journey with being a scientist, essentially, and a biocontrol scientist. So let's get straight into it. So I'm a village girl. I grew up in a small town or a small village called Pedi. Some of you may know it, people from Grahamstown. So that's where my roots arose. And as village people, you know, you're always out and about, playing outside, not on your phone, not on the screens. So we're exposed to nature from quite a young age. We've always been in touch with nature, and then that led me to pursue a career in botany, and I did that with Rhodes University at the Department of Botany, and then that culminated into me getting a PhD, looking at how we can support New Zealand in developing a biocontrol agent to a species that is problematic on their side but native to South Africa. So that was my PhD project, and then currently, as Martina has already introduced me, I'm with the South African Institute for Aquatic Biodiversity, and I also work with the Center for Biological Control. So I would say that my career has been or is multifaceted in a way, because I hold multiple roles. I am a researcher, which is the part that I'm going to be focusing on in this talk. But I'm also a supervisor, a role model, and a mentor to many students. I am also the founder and consultant at Baso R Analytics. And then let's get into the science. So Ian has already introduced the problem of invasive species, and Kim touched a bit on the problem of specifically of hot hyacinth. So you ask yourself, why should we care about invasive species? And again, we've seen the effects, some of you who are close to the Hatties situation, it can take up a whole ecosystem, and it can disperse discard the natural populations of plants and insects and fishes, as Ian has already mentioned, in those ecosystems. But beyond that, it also affects the rental properties or the property prices in general. There's been multiple studies on this, that if you have an invasion in the ecosystem, the surrounding properties also decrease in terms of value. And then you take this problem and incorporate it, or add additional stress to it by incorporating climate change, and then those effects become way more tangible and way more physically challenging. And I like a quote by Rahel, who says that climate change and invasive species are two of the most prevalent features of global environmental change. And we've seen that, and we continue to see that, that both invasive species and climate change are quite a problem. So again, why do we care about this? Because of a concept known as trophic cascades. So when you introduce an invasive species to, or when there is an invasion happening in a particular ecosystem, you find that the native primary producers get dispersed or get outcompeted out of that ecosystem. And then that also means that the fishes and the insects and the birds that depend on native species presence are also dispersed and out of those ecosystems. And I am currently working on a project that is looking at the combined effects of invasive species and climate change. And this project is looking at how we can better enable our land managers to actually be able to predict and act upon these invasions before they get to particular tipping points. So my study that I'm doing at the moment is looking at in areas where there is invasions versus areas where there is no invasions. We want to compare those trophic food webs, and also after we've compared them, so we do a control site where there is no invasion, and then we implement biocontrol, track the biocontrol changes across the ecosystem. So at 50% cover, we revisit the sites, 20% cover, we revisit again to look at how the population or the ecosystem is recovering. And we look at specifically trophic cascades and trophic food webs using stable isotopes, which are just a way to track where the food is coming from. So let's say a fish will eat a native species or a fish will eat an invasive species, then you can tell what it's been eating using those stable isotopes. And then this project is based on a transdisciplinary approach. The way in which we select our sites, we make it participatory so that we encompass a whole different range of ecosystems, and it's multidisciplinary. I'm working with people from fisheries, people, entomologists, and botanists as well. Ultimately, we want to get to co-production where we move from data to decision to impact. And the way we want to do this is by building a dashboard or dashboards for land managers and policymakers. So you remember I said we are collecting data before and after invasion and tracking the invasion. So we want to identify the tipping point in which we lose important species in those ecosystems. And then once we have those, we can turn that into a kind of a model using machine learning. And then from that model, we'll turn it into interactive dashboards that land managers can actually use. And we'll call that the Aquastar project, which is currently funded by the Jennifer Ward Oppenheimer Research Grant, which I've recently received, and I'm very grateful for that support. And just to recap, my core mission is to actually tackle ecosystem threats, both the freshwater threat and the climate change threat, by producing those interactive dashboards. And I'll give you a quick example of what we are doing. So in a recent study or a recent trip that I went on to Kruger National Park, we were working with experts across the range. So we had experts working on the algae, the plants, the microinvertebrates, the crayfishes, fish communities, the river structure, and the water quality. All of that is gonna give us information about what is happening in the ecosystem. And these are just some nice pictures. You always have some nice pictures in a presentation. So this was during our trip to Kruger National Park. And just before I close, I want to just share a little bit of the human side of a scientist. So the journey has both been rewarding and challenging at the same time. But I would say that resilience has just kept me going. My family has kept me going. I did my PhD during a season where we had COVID-19 happening, and I have unfortunately lost my grandmother during that time. I was also with a baby then. But with the support of my husband and with the support of the community and the research community I was in, I was able to push through and make it to beyond that point. And here we are still fighting. So I would say that to anyone who is keen to become a scientist, if we have any aspiring scientists in the audience, just start. Even if you start messy, if you stay the course, then you're guaranteed that you're gonna see your success, because progress is not linear, and everyone just has to start from somewhere. If you want to connect with me, you can just take a picture or scan any of these websites to follow the research. It is still in its very infancy. So if you want to know what's happening with that, you are welcome to follow me at those pages. But I'm mainly active on LinkedIn with regards to my research. So if you want to look at that, then you're welcome to. And thank you.
Thank you, Nompumelelo. Thank you so much for sharing your journey, you know, through the hardships and through the successes also. Sorry, I forgot to put my camera back on. So yeah, really insightful to just see your journey and your perspectives, and that you also mentoring the, you know, the next I wouldn't say generation, but the next series of young scientists, which we know we desperately need in this country also, but also across the world. Yeah, so we just want to be grateful for your contribution and look forward to see what you will also achieve going forward. Nompumelelo, I don't see any direct questions. There's a couple of comments in the chat that we can possibly respond to. But I think at this point, I just want to close our webinar. I want to ask Ian and Kim to switch on their cameras. And maybe we can just reflect very briefly from each of you. I've got two things that I think our audience might be interested in, and that's maybe the role of citizen scientists or any projects, so to speak, that people can get involved in, or volunteer programs, possibly. Kim, you spoke a lot about community engagement. So if there's anything you want to highlight from the institutions or the research spaces that you are involved in. And then also a question, just what new frontiers are you possibly excited about in the space that you're working in this biocontrol space? We know there's lots happening in terms of let's say genetics research and environmental DNA and those sort of things. So I don't know if any of you want to start and just close us off with your insights and in your opinion.
I can start by just saying that I think that the new exciting things that are happening, one of the really exciting things is the potential for biological control of pines in South Africa, which is a they're really damaging invasive alien plants, especially in our fynbos biome. And we're starting to get somewhere with the biocontrol of pines, where we're hoping to use a seed reducing agent to reduce their invasiveness. It's obviously been difficult because pines have been are such a valuable resource, but they're one of the worst weeds in the fynbos area. So I think that's an exciting thing in weed biocontrol that's coming up. Yeah, I think Kim can talk a bit more about how people can get involved, but it's really, it's one of the most important things is that you get in touch with us when you have a question or when you need an agent. Thanks.
Yeah, I really think knowledge is power. So I think if people spend time understanding biocontrol and talking about it and making it a normal thing, then obviously people will support it more. So really follow our Facebook page, and if you have any questions, you can email me. I'll put my email address in the chat. But yeah, we follow our pages and we try and put as much information out there as possible about biocontrol and about the newer programs and our existing programs. But yeah, and if you do have weeds that are problematic, contact us. We can help you identify them. We can help you point you in a direction if there is a biocontrol agent to it. We can't advise on chemical and mechanical removal, but when it comes to biocontrol, we know it all.
You have the
Cool.
mandate. Nompumelelo,
I'll
anything
write
you'd
my
like
email.
to add?
Yeah, just as Kim said, if you have any sites that you see like any types of invasion on, I'm especially looking for sites for my research. So please reach out to us, either to Kim or to me via the channels that I've just shared with you. Yeah.
Great, thank you. Quick questions. Is the center doing anything in the city of Johannesburg?
Yeah. So we have a as Nompumelelo spoke about, Julie Kutsia, Professor Julie Kutsia, and she works for SIAB, the South African Institute of Aquatic Biodiversity, and they have a satellite office based in Observatory in Johannesburg, and it's only been there for just over a year, maybe now. And so they are building traction and relationships with the city as well as other partners in the city. So yeah, but if you we can tell you more about that if you contact us.
Ian, and you mentioned pines, obviously the biocontrol for pines. One of our guests is asking, is it for cluster pine?
Which pine species is it?
It's pinus.
We'll direct you to the center for.
Yeah,
I'm
so
not
we have
working directly on that project.
Yeah, we have different researchers working, like specialized on different
Sure.
groups of plants. So
okay,
It attacks 3 or 4 of the most problematic invasive pines in the country. It's at least very effective on 2 of them. But I think I should leave it to the expert to answer that question properly in case I get it wrong.
Absolutely. Okay. So with that, I'm going to close off the comments and the questions for now. Thank you very much. We're slightly over time, but I just want to extend a last thank you to Nompumelelo, Kim, and to Ian for putting time aside to joining us this evening. I think we've all learned something, and yeah, looking forward to see the center grow and to see the reach that you also have in both in terms of research and your community engagement. Thank you very much.
Thank you.
Thanks, Martina.
Thanks, everybody.
Thank you. And then lastly, just to the audience, so our next webinar will be on the last Thursday of November. That's our last webinar for the year, and it will be hosted by Cherise Viljoen. So please enjoy your evening, and we look forward to see you on the 27th of November at 5 o'clock. Thank you. Have a great evening, everyone. Bye-bye.